Good evening, everyone. I'd like to thank Cesena Fiera, Chairman Piracini, Director Bianchi, and Crédit Agricole. This is a new experience for me, and I am pleased to be here and to be like a guinea pig for this new initiative, which I find very interesting. I'd also like to thank everyone here in the room, including some old friends that I'm happy to meet again. I'm going to talk about biotechnology. Don't be scared, as I'll do my best to avoid technical terms as much as possible. I'll keep it conversational and talk to you about what we do. If you have any questions, feel free to interrupt me, otherwise we'll keep them for later. So, why biotechnology? I usually start off with this slide, where I try to summarize what you hear every day, that is, emergencies, more specifically climate emergencies. By the way, what has happened in the Emilia-Romagna and Marche regions recently really saddens me. Other places too have faced similar issues, that is, periods of high temperatures, scarcity of water and drought, followed by a lot of water, floods and so on. This certainly has a very strong impact on agriculture. Similarly, we've also seen a reduction in cultivated areas and an increase in population. At the end of the day, this might not be highly noticeable in Italy, but the population is constantly growing around the world. However, what we do notice and what farmers notice in the fields are other emergencies, those that concern disease control and the plant's response to stress factors. So, how can these be managed? What tools do farmers have to be able to manage those diseases and emergencies? Some diseases are more common, such as downy mildew, while others are more recent, for instance, Scharka for fruit or Stymphalium, which is also recent, or rather, it was already known, but it has become a priority recently. Other examples are Xylella and Flavescens dorée, which we're going to cover later. What is causing these diseases? The cause certainly lies behind what is currently happening in the world. For sure, climate change has an impact on this. But some events also happen because the decisions we make can, to some extent, influence things. I'm talking about European policy and the Green Deal. Is it really the right strategy what we're offered at a European level? I mean, getting rid of chemicals, which by all means we have to use as little as possible in agriculture, but are we sure that this will equal sustainability and access to food, production, and also sustainability for our companies? In any case, if we have to tackle sustainability, environmental and consumer safety issues, we still need to have alternatives for the agricultural system. This means that removing certain substances can lead to farmers being defenseless and unable to defend their plants, ultimately forcing them to cut down the plants. This is a concept that I call chainsaw treatment for farmers in arboriculture, which is a final treatment. So, what alternative solutions can we offer? I'm not here to sell biotechnology or to say that biotechnology is the only alternative solution we have. I'm here to say that we have to look at technology and see if these technologies can offer alternative solutions that can be useful for consumers, not just for farmers, but for both of them. So, what is biotechnology? I'll start from a childhood memory of mine. My journey with science begins precisely from micropropagation. So, in vitro culture falls within biotechnologies. When I was a student, which was a few years ago, micropropagation was seen as a technology at the time. It was synonymous with innovation. I wrote about the micropropagation of kiwis in my dissertation. Then so much came after that. and the labs, and I see that there are a number of representatives of important laboratories here in the room. And now 30 years after I've first studied micropropagation, I still find limits regarding the acceptance of micropropagation. And this is also quite peculiar, if you will. Why can't we use micropropagation in strawberry propagation yet? Because we have seen variations in the past. Why can't we do it with vine? Because even with tomatoes, and seed companies wouldn't like this, it is an extreme example. Why not? When facing health problems, we need to come up with a solution one way or another. Micropropagation is an aseptic culture system with no risk of contamination. And in case there is contamination, it gets thrown away. So I use this as a first example of a cultural block that is still present in some of our reference systems. Now, everything has to be seen in detail. We need to understand how things work. I'm happy to see that several companies are now investing and experimenting in this, the tomato in this case too. As for innovation, an example of it is robots being implemented by some companies, but I already see that we will need to get to micropropagation in the future. I am not saying that everything that is now a micropropagation lab is going to be discarded, but we'll get to cell cultures, somatic embryogenesis, bioreactors, and what microbiologists do. These are all things we'll also be able to do with plants, and we will do them with plants. I think we'll get there. Fine-tuning the protocols for somatic embryogenesis means having 10,000 plants in a petri dish. It is therefore really an important system. This is the first example of biotechnology. Now we'll get to the biotechnologies you expect, the controversial ones, those nobody wants because they think they have to do with GMOs and so on. Here too, let's try to make a distinction. This slide is in English because it's taken from a project we've worked on. So here, two groups have been created, breeding and mutagenesis, which in theory are good biotechnologies, that is, traditional breeding. We do crossings, we get a variety with some improved traits, we put it on the market, and no one says a word about it to us. If it's successful, it's launched in the market. Sometimes being good at marketing matters more than breeding, which is based on crossing. Mutagenesis is a genetic variation, the creation of variability that can be induced in nature with natural tools. That is, it happens in nature. Otherwise, we can induce it using chemical and physical tools and so on. It had an impact in the past. Think about creosote, think about Mr. Lully and self-pollinating cherries. He owes a lot to what we're now discussing, to mutagenesis. And this has nothing to do with GMOs. These are good biotechnologies. Nobody has any objections. When you have new varieties, you put them on the market, and if you're successful, you can sell them. Then, on the other hand, there's another category of biotechnologies that sheds light on what GMOs and non-GMOs are, based on the terminology adopted by the relevant 2018 European legislation. These are all techniques that are based on recombinant DNA technology. We'll see this in a while. Where you see the little scissors sign, that points to recombinant DNA. What does it mean? DNA cut and sew. According to the Directive 2018, this means that in theory, any technology based on DNA cut and sew equals to creating GMOs. At the moment, it doesn't matter what sort of cut and sew we're doing according to the directive. There are actually various cut and sew options. We'll see the difference between transgenesis, cisgenesis, intragenesis, gene editing, gene silencing. We'll see it later. So what's the difference between these two categories? As I said, with the first you have something new, you register it, you put it on the market, and no one objects nor says anything about it. It might even be a variety with some degree of toxicity or allergenicity. You notice it later, but you're not asked to do anything beforehand. On the other hand, for a scenario like this, there is a legislation in Europe and a similar one in the US, in Japan, and in all other countries, but applied differently. This regulation specifies that in case of alteration based on recombinant DNA technology, you need to evaluate risks and benefits, biosafety, and prove that biosafety is there and that the environment and consumers face no risks. The main issue here at the moment is public acceptance, particularly in Europe. There's a bias. People tend to say, no, this is a GMO, I don't want it. So how do you get past this impasse? This is something we need to figure out. As I said, we'll have a look at it. And I'll also take this opportunity to show you what we're doing and to thank my colleagues here today who are helping us with all these activities. For instance, as I said, mutagenesis is a fairly simple technique. It used to be seen as the main technique in the past, but then someone discarded it in favor of gene editing. Gene editing makes specific changes possible. They are exact changes we can predict. Mutagenesis, on the other hand, involves genetic changes which we are not able to predict. They are random. What we do is we simply induce mutagenesis through chemical treatments. This is a substance, a molecule, ethyl methane sulfonate, which is used. It creates specific bonds on two nitrogenous bases of DNA, it breaks them and then randomly generates a change. I think it is actually still an important technique for vine. From what I know on viticulture, virtually all vines are the result of clonal selection, that is, the study of the existing genetic variability or natural mutations that have occurred. Vines are quite unstable at a genetic level, and therefore, if combined with in vitro culture, a treatment with new somatic embryogenesis with mutagenic treatments, will lead to genetic variability. This is what we saw in the field a few months ago. Same plants, same clone. After mutagenesis treatment, there were plants with healthy bunches and plants with infected bunches. We're obviously going to select this, which is in theory a new clone, a non-GMO derived clone. So it is a non-GMO derived variety. And even the whole variety registration system in viticulture is simplified like this. So I am still a supporter of this technology because it simplifies work. This slide is the most difficult one from a scientific point of view. I will go through it and explain it to you because it's all about this. Perhaps you have read or heard of GMOs, cisgenic, NBTs, NGTs, RNAi. All these terms, everything seems to be the same here, but it's actually not. And even in the scientific field, within the realm of technical scientific communication, there tends to be some confusion at times. What you see at the top are several pieces of DNA. What does a genetic engineer do? As I mentioned, they cut and sew pieces of DNA. Before that, molecular biologists have identified valuable pieces of DNA which determine and control traits. The red sequence at the top is the main one we're interested in. It controls traits, such as red instead of yellow, resistance rather than susceptibility, more sugar, less sugar. I'm making it a lot simpler than it is, but it's just to give you some examples. Genomics and molecular biology, the study of genomes, have allowed us to identify several genes. The vine, strawberry and apple tree genomes are known. The tomato genome we already know everything about. We know human genomes too. This means that genomics is certainly very advanced now. At this point, our knowledge of genes allows us to understand if there is an issue with a plant and whether this issue is related to the presence or absence of a given gene determining a given trait. Using the different technologies we have, we can decide to cut a gene that we don't want or insert a gene and integrate what we miss. There are different solutions here though. The first approach is saying, well, I'm missing a gene, I'm taking it from another organism and transferring it to a given plant. This approach is known as transgenic, that is the red line. I inserted a DNA sequence into a different plant. It's genetically distant from the organism I took the gene I needed from. It may even differ a lot. There is a bit of a debate on how transgenic and how different it can be, and so on. What is the point? DNA is always DNA. This is bioethics, and this is biosafety. This means that those who do this job don't do it because they enjoy moving DNA. Rather, they do it to solve problems. When this is the case, they look for benefits and safety. If there are no benefits or safety, the plant will certainly have no future. If there are benefits and safety, the plant goes on the market, as is the case around the world. To counter the debate on transgenes, the concept of cisgene has been developed. What does it mean? We create a barrier, which means that we cannot transfer DNA between organisms that are genetically too distant. With cisgene, we refer to DNA going from one plant to another of the same family through crossing, so the gene would be transferred in the same way. This is a case where it is accepted. What is a classic example? The one we're interested in, as it also represents the work done by Professor Sansavini at the University of Bologna for many years, for instance, the cisgene, that is the gene identified from Malus floribunda, has been transferred by means of genetic engineering to Gala in just a few years, thus keeping the characteristics of the same variety without losing anything of the commercially interesting variety. On the other hand, it took 50 years to obtain the first scab-resistant varieties with interesting agronomic characteristics through hybridization. So this is definitely an advantage over traditional breeding. I'll spend 5 minutes on this slide as it is the most important one, and then we'll see if there are any questions. Afterwards, we'll talk about new technologies and see some examples. RNA is interesting. We know what it is now because of COVID and vaccines. We'll go through the different RNA strategies later. After all, RNA is also important for plants. We'll see what options we have with RNA, which gives us an interesting chance to use rootstocks in fruit trees to create moving RNA fragments that make the graft more resistant. The roots are GMOs. The fruit you eat is not a GMO. This was just the first example, and we'll see others. Now, you call me to give a masterclass here today, which means I'm old, so I'm not sure I'll get to see this, but I imagine that phytosanitary products will be different in the near future. They'll be RNA-based rather than chemical-based, like perhaps most of the medicines we'll be taking. Molecules will no longer be classical, but perhaps they will also be RNA-based. Then there is gene editing. We hear a lot about gene editing at the moment. It's based on the CRISPR protein, the 2020 Nobel Prize discovery. It is a protein that is able to cut and sew a very precise DNA sequence. In this case here, even more precise. When you have an issue related to a gene, you can easily cut it. Here's the problem though. In order to make this change, you have to insert the protein into the plant. And you can't do it right in the plant, obviously, but you need to do it in a single cell. And there might be different technologies here. Depending on how the protein is inserted, you can have a GMO or a non-GMO, because if it is inserted with traditional genetic engineering, you still have a GMO. This happens when you still have the other pieces of DNA that we see above and that are needed for transformation. What are the other pieces of DNA used for? They are only used for transformation. And unfortunately, for those who do the dirty work of cell transformation, these things are needed and may be removed later. There's still a debate at European level. 15 years have passed and 3 European legislators have failed to reach an agreement on the distinction between NGT1 and NGT2. That is, NGT1 does not involve other changes. NGT2 involves gene editing and other changes. The former should be considered as a non-GMO in theory, and the latter definitely as a GMO. This is a very debated matter. This is just to show you that there are many possibilities and technologies. I would also like to mention something else related to this. For instance, patent laws. Those who deal with breeding know that once this material is commercialized, it is protected by licenses, plant rights, and actual patents in the United States. And then come the plant breeder and the licensee. These are all very well known to anyone who works in the sector. It's also worth mentioning that in addition to all this, there are now also patents for the tools used for biotechnology. For example, this molecule, Cas9, the one needed for CRISPR, that blue one there, is fully patented. So it's not like everyone can use it, or rather everyone can use it, but you need to make an agreement with the companies that hold the patents for that. But let's not name names. This is to say that this is all part of this context. Then in particular, as I mentioned earlier, if it is a GMO, regardless of what it is, it must comply with the 2018 regulation, the European one, which was implemented in Italy through Decree 194 of 2003. This has remained the same since 2003, and just like at EU level, it defines what a GMO is. So the whole technique and recombinant DNA, cut and sew concept. Then everything else, like polyploidy and mutagenesis, these are all GMOs. If you have GMOs, you need molecular genetic characterization to make the distinction I mentioned earlier, whether it is transgenesis, cisgenesis, NGT1 or NGT2. Different levels of changes in DNA might then lead to more or less important impacts in the subsequent evaluation. As I said before, if you work within this field, you first need to find a benefit, of course, and then prove that there are no risks after. And the subsequent evaluation I mentioned is precisely to exclude any environmental and health risks. So what is the approach for this? At EU level and through this directive, it's the precautionary principle. We have to prove that there is no such risk. From a scientific point of view, the precautionary principle is actually interpreted as substantial equivalence. That is, this one thing is the same as this other one, and I can therefore accept it. There's no difference. That being said, this is also a highly debated topic. Scientifically speaking, I firmly believe that we should not get rid of this legislation. I think it's a key legislation and it allows us to make evaluations and say, have a look at this, we got a result, it has benefits. That is, there are no risks or the same risk cultivating a variety instead of another, for instance. So, this was a summary of the belief or assumption that cisgenesis, transgenesis and NGT1 cannot be considered as GMOs, Well, these are actually all GMOs at the moment. As I said, no changes have been made to the European directive for the time being, and I'm not sure what will happen with the next legislature. I don't know, and I don't want to wait for another legislature and another 5 years. Let's try to proceed with experiments and prove that there are benefits and that these benefits are not associated with health or environmental risks. This is the legislation. I don't know if you are familiar with the European evaluation system and how it works, but I would like to talk about this as it is somewhat of a political topic. Who's taking decisions? It is not the researchers, because the evaluation system is political, meaning scientific and political. What does it mean? We researchers do something which is then tested at different levels, and once we have the results, we draft scientific publications. The more interdisciplinary they are, the better. This is also a bit of an issue we have within the scientific community. People tend to be self-centered, but the more interdisciplinarity you create, the better. There is an authority in Europe which is in charge of scientific evaluation, and that is EFSA. So you need to prepare a dossier with all the relevant information and submit it to EFSA. The GMO panel take care of scientific evaluation, characterization, environmental risk, health risk, in addition to agronomic evaluations on agricultural systems. A public consultation takes place and EFSA makes a proposal to the Commission. This is known as EFSA's opinion. You might think that an opinion is personal and non-binding, but this is actually the term that is used for EFSA. So what is the next step? The political choice, the authorization. In the current European system, an absolute majority is needed, so 2/3 of the member countries must give their authorization, and this has never been reached in the last 25 years, unfortunately, which is why there are no GMOs around. The last GMO was approved precisely in 2001, MON 810, the BT-resistant corn that is still cultivated in Spain and used to be cultivated primarily in Romania. This is the only cultivation example we have, which is not available in Italy. No more have been approved since then, precisely because of this political position, which is no longer scientific, as EFSA's opinions were in favor. So why? Because some are not willing to change their opinion. There is a nice book I'm quoting in this slide, Seed of Science by Mark Linus, who was one of the founders of the Greenpeace anti-GMO movement. He's someone who was beaten many times while damaging cornfields in England and Monsanto premises and so on. He then wrote a book and said, "Look, we got it, but we are wrong." So he actually understood that they were wrong, and that's really interesting. Now he has changed completely, he has started talking about climate change, but he also has to make a living, so he jumped on the bandwagon, I guess. But it was these messages that somehow created this situation. What's the interesting thing that happened in Italy? It occurred last year because of drought. It's really interesting. Because of the extremely critical situation caused by drought in 2023, the chair of Committee on Agriculture and Mr. De Castro, now I can't remember all the names, they carried out an initiative supported by a professional organization that is here today, actually, I won't mention its name, that wanted to perform an experiment or resume tests according to the concept of NGT1, that is, plants that are gene-edited, fresh, non-GMO, and so on. The page that was added to the 2003 drought decree was really important because it opened up a chance to carry out experiments in the field with cisgenic and edited plants. This happened with the first 2 fields that were made. starting with a rice field, which was immediately destroyed, showing that there are still people who don't like this. A small field with transgenic vines was created in Verona, and there you can see the president of another association working the land and planting these plants, as a proof that there is a lot of interest in this matter and we need to find a way to deal with it. We'll see, but I think we should definitely try to deal with these issues and discuss them together. Now I would like to go back to a technical topic, the difference between RNA and gene editing. Gene editing is based on that cut-and-sew protein, which can be applied not only to plants but also to animals, or better still, to all microorganisms. It allows us to make very simple corrections, identify specific sequences that can be eliminated or replaced. The issue here is that it is clearly a genetic intervention on a plant genome that interests you. And you have a new variety, and you can do that on all plants. So what's the difference? Let's make a comparison between the seed sector and the fruit sector. As for the former, since plants are self-pollinating and homozygous, you can make transformations, have self-pollination cycles, remove what we can call dirty pieces of DNA, and only leave the mutation you're interested in. That's an easy-to-get NGT1. For vines and peach trees, this approach is more difficult. F1 is not possible and you cannot do another crossing right after. So while you may have introduced a trait that you can use in your breeding program, the approach is not as immediate as in the seed industry. And in any case, you can only make changes to the genome of the plant. RNA, on the other hand, is still based on small molecules that interfere with genes. They may belong to a plant, but also to organisms attacking the plant. And these small molecules can move and can be expressed directly in plants, which means we can have MGO plants that have a stable expression, meaning that they create molecules and make them resistant to diseases, for example. We'll also see how they can be used to create also new formulations, as discussed before. We really like RNA, and we have been very happy recently with the third Nobel Prize being given to RNA, proving that there really is a lot of interest. Our work is related to the first Nobel Prize, that is, the idea that Fire and Mello developed with the gene silencing mechanism, which also exists in plants, and we are therefore working on. Last year's Nobel Prize did not concern silencer RNA, rather RNA messenger, so genes were not blocked, they were activated instead. This was to induce our immune system to protect us against COVID in this case. This is not something we can do with plants, as they do not have an immune system, so this approach clearly does not work with plants, unlike the latest third Nobel Prize that was awarded 2 weeks ago. There is also a connection with the first Nobel Prize, as we're talking about small fragments of RNA moving around and having a regulating effect on the plant. There is an interesting article by Anna Meldulesi in Le Scienze, which is, I think, a good rootstock graft example. Pretend you're on a bridge going to Manhattan and there are cars going both ways. On one side you have RNA sending signals towards the leaves, whereas on the other side you have cars going in the opposite direction and sending signals to the roots. Until a few years ago, it was not clear why the plant's response was different based on the rootstock graft combination. I think that knowing this opens up opportunities beyond medicine, as this has really a lot to do with medicine and makes you understand a lot. I'll now move on to some examples of our work, plants and formulations. An advantage of RNA is that, going back to the possibility of silencing genes, we've already seen that we're able to identify the target genes we don't like. Whether we want to block pathogens, viruses, a fungus, we block the replication genes of that virus or fungus. We can do it with the interfering RNA approach. We can do it by expressing RNA in the plant, thus making it resistant. Or we can create, study and synthesize these sequences. Here too, COVID has helped, if you will. Everyone started to produce RNA because we needed it. That was a different context, obviously, but this is how many companies expanded. And here is where I can quote the title of my presentation. We have to invest in biotechnologies. There are no European or Italian companies doing this, so any business starting to think about such solutions is more than welcomed. If not, you'll see that some companies will take over. This slide shows how RNA works. I wouldn't bother going through it. We can talk about it if you want. But the work we are carrying out is precisely to identify target genes and prepare constructs that express the sequences, those at the top. They are specific to target genes, and the mechanism that cuts such sequences make them move, and these small fragments interfere with and block given genes. These mechanisms can work on the target genes of plants and pathogens. So our reference is still strawberries for certain aspects, but vine is turning into one as well, with the purpose of having vines that are resistant to diseases. This is where we are now, and there are people saying, no, don't show photos, don't disclose where they are, don't show their location, because then everyone will go there and destroy the greenhouses. And I'm looking at the two people concerned here. But I say, no, let's do it. If they destroy a greenhouse, then we'll make another. That's not a problem. If a rice field gets destroyed, then whatever. We'll plant another rice field, or 2 or 3. I don't know, but we have to see and do these things. And the grafts here, the idea is to test mobility from rootstocks to grafts. That is, to have rootstocks that have a stable expression of RNA moving upwards and protecting the plant. This is the work we're doing. Then we have Luca here, who follows directly, Capriotti and Casala, the whole group. We already have the first results. Firstly, we show that with the rootstock, RNA goes upwards from the roots, and that's clear. Secondly, using rootstocks provides better protection compared to varieties modified with lots of RNA. It seems that there is even a balancing mechanism, which means that the silencing system becomes saturated if there's too many target RNAs. So having a controlled expression is definitely interesting. This is the work on drupaceous fruit for Sharka. In this case, we have created 2 sites, and we'll see what happened later. The results seem interesting. At the moment, I'd rather not share much about them because we are currently carrying out further analysis. We have done more sampling recently with a Center for Nursery Activities that assists us to see what happens. Using a rootstock that's been modified to be resistant to Sharka with RNA mobility seems possible. Not only are the varieties resistant to Sharka, they don't have it at all. We'll see what future data shows, but it seems that grafted varieties are giving very promising results at the moment. I also wanted to add the work we did in cooperation with Innesti Leopardi on this slide, although the colleagues are not here today, unfortunately. Using the same approach for tomatoes is nicer and more interesting because the results you get to see are also very interesting. We don't have a project for tomatoes, unfortunately, but we managed to find someone that supported us with this. We have the first line, so we'll see in the future. It's the same concept, though. You can modify the roots to make them resistant to a range of pathogens, botrytis and downy mildew in this case. I'm focusing on RNA here, but there are also peptides actually, which are small molecules sharing the same function. And why are peptides interesting? I get people coming to me and say, so now we can solve the Xylella issue. Well, no, the Xylella issue can't be solved with RNA because bacteria are less evolved organisms and have not developed the RNA gene silencing mechanism, so it won't work. So we need to use another strategy to tackle Xanthomonas, Xylella and Flavescens doré. In that case, the strategy involves small peptide molecules that can be identified in nature or generated through synthesis, like aptamers, which are synthetic, even though they're slightly different as they are molecules. So what are we doing from here? We're taking care of the needs that have been identified. This is why I say we have to respond to emergencies, try to understand what the emergencies are, and see if this strategy works. So this is how this project was born. How can this work? Like here, for example, Stemphylium is an issue. We have to understand if the sequence of the Stemphylium genome is there. It's quite easy with fungi. The work here is done by Professor Beraldi's colleagues at the University of Bologna to identify the genes of a pathogen. These might be target genes, which create problems for the pathogen. Sequences are identified, RNAs silencing these sequences are generated and screened until you find the most interesting sequence. This is the same approach we're taking, that is, a stable expression in the plant or the formulations example we're going to see later. This is an old slide. There have been steps forward and we have an upcoming meeting to check updates, so we'll see what's happened. Another example is Flavescens doré. This is also an issue in certain areas and with pathogens like these, when you have both the pathogen and the vector. There are actually two things you can act on. The vector target, that is insects, Scyphoideus in this case, and the specific target, pathogens, phytoplasmas here, which are not evolved enough to have RNA. Insects can have RNA though, so there are on the market. Later we'll see an example of the first product created resistant to Colorado potato beetles, which is certainly very interesting. So this RNA technique works with insects. It doesn't work with bacteria or phytoplasmas, which is why we think of peptides being interesting proteins. Let's not forget about strawberries, as they are a reference in our work model. And here are the first results. The work that has been published shows that by expressing RNA in the plant, you can reduce and block infections, therefore making it more resistant. This is not a rootstock. These are actual GMO strawberries, so we take them as they are. Another work we are carrying out is once again a collaboration with the University of Ghent in this case. Here too, phytosanitary products are helpful in order to develop resistance to Drosophila. This is another emergency we have to deal with, and because we have so many, the result for the Drosophila Suzuki case has not been validated yet. But we have found the plants and we are working on it. I'm also thinking about editing cherry rootstocks and try this on cherry trees too. It could be a solution where Drosophila is an issue, but as I said, there is already a lot to do. I'm nearly finished with my presentation. Just a couple more slides. In genome studying, what is an important trait to improve plant adaptation? As for strawberries, these are the low requirement for cool temperatures and reflorescence, meaning genes that can induce flower differentiation independently, without the need for call induction or fewer daylight hours. The reflorescence trait is fundamental. Now everyone is looking for remontant varieties or low-chilling remontant varieties because they can be cultivated in several environments, and it's not just for strawberries. This work was also published. Now we have also gone on, and thanks to the same collaboration I mentioned, we've been able to identify the strawberry genes that induce flower differentiation regardless of daylight hours. This is exactly the reflorescence trait. This has been an experiment where we tried to overexpress a given gene or to silence another, so both approaches are possible. But then what happened? It was too much. Overexpressing gene S1 resulted in changes to the plant, which becomes too remontant. We have to modulate the expression of this gene through promoters for balance purposes. The plant we used no longer made stolons, but it only made flowers. This approach would be considered cisgenic, because we have the strawberry gene, the strawberry promoter, and also a resistance marker that has been identified in strawberries. So again, this is a cisgenic approach. The next slide focuses on kiwis. I kept it in the presentation even though it does not concern us, but I would like to mention my colleagues in Udine, Professor Cipriani. This is also another important goal, that is, obtaining self-pollinating kiwi plants by editing sterility genes. Here are the first results. I hope they'll really have a chance to go ahead with this experiment, which is extremely important. Coming to the end of my presentation, as we are in Emilia-Romagna, I'd like to dedicate this last part to this event that took place 2 years ago, where a number of representatives of the Emilia-Romagna region were present. I never really understood how this was possible, but the president of the Emilia-Romagna region and the councillor for production signed a collaboration agreement with a biotech company in New York, and I thought this was very interesting. It opened a path for collaborations, and I believe that we should do more about this. That is, we shouldn't leave it in the hands of this biotech company alone, which has faced issues in the meantime, but is still in business and we're still working with them. They were able to develop the first RNA formulation that has been already approved and is being used in the United States and in Australia. And thanks to the collaboration that started from the meeting with Astra, they have been carrying out experiments for 2 years and have been able to prepare a dossier to request the authorization for sale, application and use of Calantha in Europe, which is the first commercial product that is resistant to Colorado potato beetles. So this is very interesting. Why resistance to Colorado potato beetles? Salespeople understand it right away. If you make an investment, where do you make it? You do it where you are sure you have a lot of money coming in. Taking agriculture into account, what is one of the main crops? Potatoes. They're a basic commodity. So having a product that can be used for potatoes, registering it in different parts of the world is certainly a good investment. Differently, having a similar result for pear trees would be more complicated, as they're not as important. But in any case, we managed to convince them to help us with botrytis, and this is the pre-registration work. So what's the concept? It's the same thing. You have a pathogen, a gene of interest to be silenced, you find the sequences, you test them, you do the pre-screening, and then later find the most interesting formulation. Here is the example. This is the non-treated one. It's clear that when we make a comparison with traditional fungicides, they protect more and are more effective. But they're chemicals. At one stage we said, some products are also allowed in organic farming, so why not compare them with these? Some RNA formulations are currently equal to those allowed in organic farming, so why not? The authorization they have obtained in the United States and Australia actually falls under the pesticide category, so they are phytosanitary products, and this is what we should name them. As you know, there are two categories, chemical and biological. The former is chemical pesticides, which we all no longer want. The latter is allowed and can be used for organic farming, also in the United States and Australia. These new formulations with Calantha have been classified as biological pesticides, which I believe opens up new opportunities. There might still be people who are not in favor of using RNA, and that's okay, we can accept it. But as a product, it can still nevertheless be equated to a natural one, with what I consider to be a very specific target. Let's take another example, copper. Now, I don't want to say copper is bad. It's a natural chemical molecule, but it's also a chemical molecule with a wide range of uses. Whereas RNA, in this case, only silences a given gene that has only a given target pathogen, not other pathogens or possible organisms linked to it. More or less the same result was also obtained in vines, and we are gathering data. For what concerns pear trees, the work on stable expressions in the plant has gone on, and the formulations produced have been tested. In this case, we had an issue with toxicity. Too much RNA doesn't work, so we really have to learn to get doses right. I don't know, perhaps I've tired you out. We got to the end of my presentation, so now let's draw some conclusions and then I'm available for any question. Integration is really needed, and scientists don't belong to laboratories exclusively. They have to speak. So I'd like to thank you for inviting me here to share my ideas with you and with the professionals of the sector, so companies, farmers, and so on. Politics is also something we should address. You've seen that I mentioned a directive at EU level earlier and how it is applied. It's also a decision at EU level, and I imagine it is also influenced by public opinion. So if we talk about it, there's a chance we'll get what we want, as was the case last year with the drought decree, which was drafted by two politicians and allowed it experiments in the field, something which until then was not possible. Anyway, this is what we need. We also need to be allowed to do experiments in the field. I don't want these to be just words. I also want to show concrete actions, and I hope I'll be able to show some ideas we are currently working on at Macfrut next year. There must be an indisputable scientific base, so we have to be able to do our job, and this needs to be clear to everyone. Then, based on this, we can gather consensus within institutional bodies. Multinational corporations exist, and we cannot stop them. They move forward. But the issue is that if we do not move forward, they will. And if we stop, we lose control and we really lose the ability to take action, even at a small scale. It might not be the case for potatoes, but it can definitely be for pear trees and vines, which are not as important. So this is the state of things and how the world is. This is globalization, and I don't think I can stop it. But we have to be able to make our contribution. Among the various techniques we have, I'm leaning towards RNA at the moment. Gene editing plays an important role, but I think that we as researchers have to cooperate with those who need assistance and solve issues together. It's like when you call a plumber. You do it because you have an issue at home and you need something fixed. And only a plumber has the right tools to fix that issue. So here too, we need the right tools, and we need to be able to choose the one that works better. Results have to be seen firsthand, and I really stress this. As you can see from the last bullet point, we need funding, not only for research, but also from companies. I would really like to see investments from companies. Some are already showing interest and are investing in all of this. But we also need to have a strategy. I'm not saying it has to be nationwide, maybe it's too much, but at least we should think of it as something that we try to carry on doing, because it can help us, and we know it will help us solve issues that would otherwise be difficult to solve. So this was my message, and I hope you find it interesting. Thank you very much for your attention.